All right. Next thing. Let's take a look how this pathway works. It takes a number of steps, and what I'm going to do is I'm going to both kind of write the thing out and do a flowchart diagram of this pathway. Now, by the way, how all this stuff is posted up on Blackboard. It's been up there for about a week or so. Okay, so let's take a look at this. Okay, first thing we have is a receptor, a growth factor receptor in the membrane. Okay, and like all receptors, it spans through membrane completely. So here's the receptor, we call it R. And now a growth factor binds to it. Okay, that's step one. Now when that happens, that causes a structural change in the receptor. And now the receptor is able to grab on and bind a second copy of the receptor. In other words, it can stick to another receptor at the same time. So, second receptor binds. Okay, so now we got a second receptor, and that second receptor also will bind to the growth factor as well. The same receptor to the same molecules. So now the receptors are bound together, and they're bound to the growth factor. That causes yet another structural change in both receptor molecules. Because it turns out, these receptors are protein kinases. In other words, they're enzymes that phosphorylate other proteins. But they're not your garden variety protein kinase. Most protein kinases stick phosphates on the amino acids serine and threonine. This one sticks sticks phosphates on the amino acid tyrosine. So it's what we call a tyrosine protein kinase. And when they were first discovered, it generated a great deal of interest because a lot of these seem to be involved in the biology of cancer. They didn't know how or why yet, but we have a much better idea now. So what happens is when the two receptors bind, it activates the kinase activity in both of them. They're the same molecule. So now they're both active kinases. So we'll say tyrosine kinase activity activated. Just by the two receptors binding together with the growth factor. So that's the next step. Now, these are active kinases. They phosphorylate target proteins. You know what their targets are? It's each other. They phosphorylate each other. So this one here sticks phosphates on tyrosines of this, and this one, meanwhile, returns the favor. Okay, you stuck a bunch of phosphates on me, I'm going to give you a taste of your own medicine. I'm going to stick phosphates on you, too. So now the receptors phosphorylate each other. So that's step 4. Now, what does that phosphorylation do? It actually does not do anything to the receptor itself. But it turns out there is a class of adapter proteins that bind to these phosphotyrosines. These adapter proteins fall in the family called the SH2 family proteins. And there's a whole bunch of them around. But the main thing is that these are adapter proteins, and they all have a structure region that's called the SH2 region. So they're all relatives of each other. But these function as adapter proteins. And what they do is they will bind to a phosphorylate receptor, but they won't recognize one that doesn't have the phosphates on it. So we'll say SH2 proteins. Now the SH2 proteins, when they bind the receptor, they undergo a structural change, and now they can regulate other types of proteins. So, So in this case, the SH2 proteins play that role of those intermediaries that recognize the receptor itself. Notice the SH2 proteins are not G proteins. The G proteins get activated later on. Okay, so we've got our SH2 family proteins activated. Okay, they can do a number of things, but for our purposes, the main thing they do is they're going to bind to a guanine nucleotide exchange factor, or GEF. Now recall, these GEFs are proteins that bind to and stimulate GDP for GTP exchange in G proteins. In other words, the GEFs activate G proteins. When we look at the phosphoinositide cyclic nucleotide signaling, in that case, the receptor itself directly bound and activated the G protein. So it was a GEF. In this case, the receptor has nothing to do with that. So we activate a GEF. That's our next step. And like I say, the SH2 proteins can activate other things as well. Okay, GEF, guanine nucleotide exchange factor. Now, that's activated. It can bind to the appropriate G protein and stimulate the exchange, thereby activating it. And the G protein target here is our little old friend Ras. Okay. What the GEF does is stimulate Ras to expel GDP, pick up a fresh GTP, and thereby activate. Now, Ras is a monomeric G protein, single subunit one. Now, what I did here, I draw it as a circle in the inactive state, and the little squiggly line represents a covalently bound lipid that anchors it just underneath the membrane. The protein itself is quite polar, will not go into the membrane on its own, but with that lipid anchor, it keeps it at the underside, the cytoplasmic underside of the membrane. That's where it generally resides. Okay, so our seventh step. Binds and activates that G protein Ras. And there's a couple different versions of Ras around, like 3 or 4 different versions. Okay, so now we've activated Ras. We're going to represent that by turning Ras into a square and having GTP in the guanine nucleotide binding site. So Ras has undergone a structural change. It is now active. And that means it can bind to and regulate other target proteins. So what's the target protein here? Okay. What it binds to and activates is a small family of kinases called RAF. That stands for Ras Associate Factor, because when they were first looking at it, they saw that Ras bound to it and did something to it, but they didn't really know what the stuff did, so they just called it a factor. When you hear the term factor, it means nobody knows what it does. That's future work. So let's put the RAF kinases here. These are protein kinases. So now we're starting to get the first set of kinases get activated. Now what do these guys do when they're activated? They phosphorylate and activate, and I'm going to put P's here for phosphorylation. They activate a second set of kinases. So RAF phosphorylates and activates a set of kinases abbreviated as MAPKs. We'll see what that is in just a moment. But for the time being, until we finish this bit up, right? These are a set of kinases, a family of kinases. So now they're activated, they can go and phosphorylate other things. And what they do is phosphorylate and activate yet a third set of kinases. And these are called the MAPKs. So, step 10. Once we activate these kinases, then they're going to phosphorylate numerous target proteins, including things like transcription factors, in other words, gene-regulating proteins, proteins that regulate the cell cycle, stimulate cell division, things like that. And that gives us our final cellular response. Now, we have here, you notice we have a three-stage amplification here. We activate one set, a small family of kinases, the Ras. Each one of those can phosphorylate and activate a whole ton of our second set of kinases. And then they go out and activate an even bigger pile of our third set of kinases, the MAPKs. That's where the pathway comes from, the name of the pathway. So by the time the end of it, you've got thousands of these guys swimming around, phosphorylating everything in sight. Now what that means is a little tiny bit of growth factor goes a long way in terms of cellular response. Tiny, tiny amounts, nanomolar, picomolar, even less, can do dramatic changes in cell function. It can cause a cell to proliferate where it ordinarily wouldn't. It can cause a cell to turn into a more specialized cell type. And because we have this massive amplification here, we now end up with a situation where a little tiny bit of growth factor has a big whopping cellular impact. Because we've got a triple stage amplification here. Now, by the way, what does all these abbreviations, MAPK, okay. Mitogen associated protein kinase. Now, what in God's name is a mitogen? A mitogen is something that stimulates mitosis, cell division. So when they noticed cells dividing, they found these molecules that seem to be activated in dividing cells. So they said, when you added something that stimulates cell division, you saw these particular proteins active. So they said, mitogen associated. And then later on, they realized that these things were protein kinase enzymes. So now you get mitogen-associated protein kinase. Now this one, MAPK kinases, because they phosphorylate and activate the MAPKs. So there is method to the madness of all this kind of all these abbreviations and acronyms and things like that. Okay. We see numerous, yes? Phosphorylation of targets. Yeah, okay. In other words, we activate the MAPKs. Now they're going to phosphorylate all kinds of things. Special targets include things that regulate the cyclin and CDKs that stimulate cell division. Other targets, transcription factors, proteins that are going to turn genes on or off. So, you do all kinds of things with these guys. Okay, well, second thing about this pathway, it's critical in all kinds of developmental events. You see FGFs, especially in all kinds of tissue formation, limb formation, not the only thing, of course. Lots of organs, all kinds of mesodermal derivatives. You find FGF members playing roles in that. EGF plays a lot of roles too. Of course, your central nervous system and differentiation and neurotrophins are critical on that. So we're seeing all kinds of developmental events over and over and over again that ultimately use growth factors that fire off this pathway. And when cells get damaged, if you have a wound, if I were to take out a penknife or something and kind of dip it out of my arm, don't worry, I won't do it. I don't like the sight of blood, especially if it's my own. What would happen? The cells at the edge of the wound would realize that they're not attached to any other cells on one end. that would stimulate production of various growth factors and would stimulate those epithelial cells, at least cells in the dermis, to start dividing, to fill in the wound, to start differentiating, to turn into the different layers of skin. And after a period of time, I would have that wound filled in. I might have a little scar, sometimes get a little slight overgrowth, but I would have that scar healed. I'd have that wound healed. And then, having filled in that wound, healed the wound, they'd stop producing growth factors, the division would stop. And now, you're fixed up. Same thing with all kinds of organ damage and things like that. Same kind of thing. So a lot of tissue repair, tissue maintenance, development, all kinds of developmental events use various growth factors. Many of them use this pathway. Now, needless to say, if things go really wrong with this pathway, that is an extremely bad thing. Because if you misactivate this pathway, it means you're going to be stimulating cell division when you really don't want that to happen. That's what we call cancer, abnormal cell division. Anything on this pathway, if it goes wrong, is a potential stimulator of cancer. Probably half of all human cancers involve defects in this pathway, where it's overactive or active when it's not supposed to be active. But it turns out there's about 3 major hot spots that are frequently prone to mutations that stimulate abnormal cell growth. So let's take a look at that, because there's a lot of interest in knowing everything down to the atomic level on how these pathways work. We have tertiary structures of most of the proteins in this pathway. And we have a good idea how to regulate. Because that is going to be very important. Because that way, if you have those structures, you can design drugs that can bind to those structures and inhibit it in one way or another. And we actually have a few out in the market now, and more will be in the pipeline. The sooner the better as far as I'm concerned. Okay, well, so let's look at a couple cancer hotspots in this pathway. The worst is right over here. Our old friend Ras. Nearly a third of all human cancers have Ras mutations. For some reason, common mutations involve losing one of the part of the N-terminal or front end of the Ras protein, or the gene for that. Unfortunately, that front end, that N-terminal end, also contains the GTPase active site. So, if you lose the front end of Ras, what happens is the protein can bind GTP but cannot hydrolyze it. What that means is once you activate it with GTP, it has no way of shutting itself off. You switch it on, and there's no off switch. So you turn it on, it continues long after you want. And it continues firing off the rest of this pathway. That is a bad thing, folks. Now, there are backup systems. When you come down to it, in general, no single molecular defect will turn an ordinary cell into a full-blown nasty cancer cell. We have all kinds of backup systems. If something goes wrong, there will be a backup that will ramp it down. But if you start losing too many backup systems, then it can push you over the edge. And of course, that's also a function of aging and things like that. The older you get, the more prone to cancer you are and stuff. Okay, so generally, at the minimum, it takes 2 or 3 specific molecular defects to turn a cell into a cancer cell. Most of the time, it takes more than that. So we have all kinds of different backup systems and redundancy to prevent that from happening. But sometimes you overwhelm all that, and now you're in trouble. Okay, so Ras mutations are a biggie. That's found in almost a third of all human cancers. That's a lot. And then what happens? You lose the GTPase activity. It means you turn it on, you can't shut it off anymore. Second thing, some of those receptors, specifically the Egf receptors, they're bad boys in this regard. Because certain EGF receptor mutants, what will happen is they can actually bind to each other, activate the respective kinases, and phosphorylate each other even when there's no growth factor around at all. So in other words, they switch themselves on without growth factor. Bad idea. Because growth factors are produced and released and tell cells, why don't you start proliferating? When they start doing it on their own, even without a growth factor, that's a very bad thing. There are drugs, Herceptin is an example, that's used to treat a number of kinds of cancers, including certain types of breast cancer, that act to bind to and inhibit the EGF receptor. It blocks it and tries to ramp it down, keep it from doing what it's not supposed to be doing. So that's out in the clinical market now. Another thing found in a certain kind of cancer are good old RAF kinases. One member of the family, called RAFB, is found in the majority, 70, 80%, maybe more, of melanomas. Turns out that RAFB mutation allows it to activate the rest of the pathway even when there's no RAS around. It switches it on permanently. Bad idea. Now, of course, melanomas are some of the nastier, quicker, more aggressive cancers you can get. There are cancers of these cells, these pigment cells called melanocytes. Now, the thing with them is these cells are not like ordinary skin cells to begin with. They're derived from this tissue in early development called the neurocrest, cells that have kind of like halfway between nervous system type cells and between mesodermal cells. In effect, one thing that neurocrest cells do when they're first formed in very early embryonic life is they proliferate, spread all over the organism, settle down places, and turn into all kinds of things. They turn into the pigment cells of the skin, the melanocytes. They turn into part of your jawbone. They turn into certain endocrine glands. They turn into your peripheral nervous system, a number of other things. Problem is, when these kinds of neurocrest cells go bad, they revert back to their childhood. What they do, spread all over the place and proliferate. It's fine in development, but when development's over with, it's a really bad idea. So they tend to be neurocrest cells. Neurocrest-derived cancers are very bad cancers. Think of melanoma. Think of glioma. That's what took down Ted Kennedy in a year or so. Those are some of the nastiest, hardest to treat, and quickest to cut you down types of cancers. Melanomas, many of them have these mutations in RAFB. There are right now on the market 2 recently FDA-approved drugs that bind to, and specifically, I think they bind to the active site and inhibit RAFB. It's not a cure, but it does extend it does extend life. So, here again, a lot of interest in this pathway and many other signaling pathways, because when signaling pathways go wrong, that can stimulate cells to go into a cancerous state. Now, I like to put one more little thing. This is a little historical aside here. We don't have that time to cover another pathway. But a little bit of kind of interesting history here about we've seen when good signaling pathways gone wrong. Well, you can have science gone wrong, seriously wrong in this case. We go back into the late 1970s. People were starting to map out aspects of some of these growth factors and how they worked. And it was a super competitive field because, of course, with a lot of the top labs, cell biology and biochemistry labs in the nation working on these, of course, we knew that these growth factors were involved in embryonic development. They stimulate cell proliferation. So automatically, there's a potential connection with cancer and stuff. And that means if there's a connection with cancer, you can get lots of grant money from the NCI, right? Perhaps not as much as it should be out there, but it's a hot field. Well, go to Cornell. There was a very prominent, I think he's retired now, biochemist named Ephraim Racker. He was working on this along with many other people. And he had this new grad student, a guy named Mark Spector. He seemed to be really just an absolutely brilliant type of person, the kind of person who might be able to write his own ticket when he finishes up with his PhD. And Spector had this idea that somehow those growth factors bound the receptors and somehow stimulated a chain reaction of protein kinase. Turned on one set of kinases, they turned on another set, and so on down the way. And they got a massive cell response from a very tiny amount of growth factor. So, great hypothesis. So he starts doing some experiments, and it starts indicating that, yes, that's what happens. You fire off a chain reaction of protein kinases. And he was getting like first author papers in the best cell biology journals and stuff like that. He'd go to a conference and present his stuff, and people could hear a pin drop. He was like, wow, what's he come up with now? Guy is looking like he is hot as a firecracker. And then other people start trying to do the same experiments to replicate the results. Now we start running into problems. They couldn't get it. They couldn't get this stuff to work. So, all right. Rackard calls up Spector and says, hey, let's see. I've got a bunch of people saying I can't get this damn thing to work. Why don't we go and repeat these experiments? I mean, yeah, sure. He goes out, repeats experiments, gets the same results. Great. It works fine. So, okay. So, yeah, I got it to work again. So, still more people in other labs couldn't get it to work. This is looking pretty nasty. So now, finally, Racker himself goes out of the office and into the bench and starts trying to do the experiments himself. And he's a really good biochemist. Well, he could only get to work when he was working with Spector. When Spector wasn't around, even he couldn't get the stuff to work, even though he's following exactly the same procedures and protocols and stuff like that. So things started looking really, really suspicious because Spector was the only person to get this stuff to work. And nobody else could. That makes things look suspicious. So eventually, he got the boot out of the lab, and then Cornell's PhD program gave him the boot out of the program, and he disappeared into obscurity. Nobody knows how he pulled it off and what he did, but clearly he was cooking the books in one way or another to support his hypothesis. The ironic thing is, his hypothesis was right, he wasn't coming up with evidence for it, so he decided to make his own, however he managed to pull it off. Years later, other people using other techniques did indeed find that that pathway triggered off the cascade of protein kinases that we just went through. So at this point, it's a good time to hang it up. I will see some of you folks live. Don't forget, when you go to lab, turn the spectrometers on if they're not already on. Make sure to set 475 and fill the switch is in the right place. And